My name is Yi Wen. I'm a PhD candidate in the Iru Dayaraj lab. I'm from the Department of Engineering, Bioengineering at UIUC. So we all know that perfluoroalkyl substances are persistent environmental pollutants that have significant adverse effect on our health, with the aim to mitigate this chemical's toxicity and to validate the outcome on a biological system. Our collaborators and I first defluorinated PFAS and then tested the chemical residues on a novel in vitro liver model. So to achieve the defluorination, PFAS were treated in a closed system photoreactor as shown on the left-hand side. This system includes a mercury lamp emits UV-generated hydrated electrons. And also multiple concentrations of PFOA and PFOS with different treatment time were tested. And in our conclusion, we found out that 24-hour treatment was sufficient for the defluorination of either PFOA or PFOS at concentration equal or lower than 100 micromolar. But we also found that in the residue, in the defluorinated residue from mass spec, that partially defluorinated products do exist. So we think it's important to investigate the biological influence of those residue compounds. Single cell assays are often used in vitro studies. However, liver is composed of many different types of cells, and we know that cell and cell interaction as well as the signaling between cells can significantly influence the cell state, therefore impacting the susceptibility of to toxins. The left figure shows a prototype of our co-culture dish, and the diagram on the right-hand side shows how it was made. So in our co-culture system, we are culturing the hepatocytes, which is the HepG2 cells, and the hepatic stellate cells together. Hepatocytes, HepG2 cells are cultured in a 3D spheroid format on the gel pad in the center, and the hepatic stellate cells are cultured in the surrounding area. After culturing, we can also separate two types of cells and use them separately for analysis. So we think that our system will more closely resemble a liver than other in vitro system, but this can be used to better control the environment, and the assays can be done more easily than in vivo system. Last, I would like to show you some of my preliminary results. I've chosen to show the HepG2 cell gene expression on CCNB1 and CDKN1A genes. CCNB1 encodes an essential cyclin that controls cell cycle phases, and CDKN1A may be involved in the inhibition of self-proliferation responding to DNA damage. And both proteins are essential in controlling the cell proliferation. In the first figure, we can see that after the PFOA treatment, the CCNB1 gene expression has decreased significantly, and after the PFOS treatment, the CCNB1 gene expression has significantly increased. While in our defluorinated PFOA or PFOS treatments, there is no significant difference between the gene expression between them and the vehicle control.
1 minute.
And for the gene expression of CDKN1A, we see that PFOS has significantly increased the expression of this gene, and again, the defluorinated PFOS has no significant difference between the gene expression of this gene and between the defluorinated PFOS treatment and the vehicle control. And this project is still ongoing. That's all I have for you today. Thank you.